base on this because I know we ran out of time and I know it's part of your content. Okay. So I want to ask you all, what did you all find was the most likely cause of the cyanide poisoning? It was the Tylenol, right? Somebody had tampered with the Tylenol and caused, cyanide poisoning in these few individuals here, right? So this was a real case, actually happened in Chicago. I can't remember the year. Many people died from this, and who did it? Well, we don't actually know. They never caught the person. So that person got away with it, but silver lining of the situation, these murders is what actually caused a lot of these seals. So, if you see the sealed for your protection or do not take if the seal is broken, right? That came about because of these murders, right? We had to have some way to see if anybody tampered with the product you're buying. Any questions on that cyanide murders case study before we move on? All right, we are going to transition into one of my favorite topics because I'm a plant person, photosynthesis. So first of all, plants use cellular respiration as well, right? So we talked a lot about cellular respiration. Plant cells are going to use the same process, same pathways involved, right? We get the glucose that goes through those pathways from the food that we eat, right? Plants have the ability to make their own glucose, which can then go through cellular respiration. We'll see that photosynthesis produces ATP, right? But think about all the cells in a plant, right? Not all of them are going to be photosynthesizing. Some of them are literally underground in roots and do not have access to sun. So particularly roots, right, will really rely on cellular respiration for their ATP. All right, so I kind of mentioned this before we started cellular respiration, that it's a cycle, right? If we look at cellular respiration, we are taking in glucose molecules plus oxygen, creating ATP, but the byproducts are water and CO2, right? Photosynthesis uses that water and CO2 to produce organic molecules like glucose, right? So it's a cycle. They kind of switch between both of them here. They play in really nicely with one another. In the process of photosynthesis, we will see that we are actually getting oxygen, right, which we rely on, lots of life on Earth relies on, right? So think of it as this constant cycle. Some of the outputs from cellular respiration will be used in photosynthesis. Some of the outputs from photosynthesis will be used in cellular respiration. And not necessarily in the same organism, right? Think about we are exhaling the CO2 that plants might be able to harvest and go through photosynthesis. We take a look at the overview of photosynthesis, right? What is happening here is that CO2 is being reduced and water is being oxidized. When that happens, we are producing sugar, glucose, and oxygen. This reaction is not energetically favorable. If we look at the Gibbs free energy here, it's positive 685. So this reaction does not happen spontaneously. We need an energy input here, all right? And that energy comes from light. It comes from the sun, right? So I know we've talked about if you need an energy input, it's typically going to be ATP. But in this case, it's from the sun. It's sunlight that provides the energy to make this reaction go. And it provides a lot of energy, right? This is a plus 685 Gibbs free energy, right? That's hugely endergonic. It's not going to happen spontaneously. So the sunlight provides a lot of energy for this to happen. Right, and again, that generalized equation, CO2 plus water equals glucose and oxygen. All right, like I mentioned, not all organisms can do this. So we are heterotrophs, right? We have to eat food to gain our energy, right? There are autotrophs, so individuals that can make organic molecules from inorganic sources. Right? This can be from sunlight, in which case we would call them photoautotrophs. So if they make their organic molecules from the power of sunlight, they're a photoautotroph. There are also things like chemoautotrophs, which uses chemicals in the environment to make their own energy. I believe a lot of like deep sea, like bacteria and organisms rely on some like chemical geysers in the ocean to provide them with energy. Much less common, right? But they do exist. So photoautotrophs, again, we're talking about anything that photosynthesizes. That could be kelp, cyanobacteria, which are small single-celled organisms, plants, some protists, right? You like euglenias, they can photosynthesize. Whole variety of organisms here. In fact, cyanobacteria was thought to be one of the first photosynthesizer and where we actually get chloroplasts from, right? Because chloroplasts came from that endosymbiotic theory. It was thought that it was once a cyanobacteria. All right, in order to understand where this is happening, we have to understand some basic leaf anatomy. All right, so if we look at a leaf, and this is if we took a leaf, cut a chunk out of it, and kind of looked inside of the leaf, right? This top portion here is the top of the leaf, and then the underside of the leaf is hidden in this image, but it's underneath this yellowish layer, right? The majority of photosynthesis occurs in a specialized cell type called the mesophyll cells, right? There's actually two types of mesophyll cells, but I am broadly generalizing it to mesophyll cells for this class, right? We can have this layer on the top, we call palisade mesophills because they're elongated, and then the layer beneath is a spongy mesophyll. They both contain chloroplasts, right? They're both going to be used in most of photosynthesis for the entire plant, right? So mesophyll cells are the majority of cells that are in between the top and the bottom of the leaf. If you were to cut open a leaf and randomly point at a cell, chances are you would be pointing at a mesophyll cell. They're that abundant. We also know we need carbon dioxide, right, in order for photosynthesis to work, right? We're combining carbon dioxide and water to get glucose and oxygen. Well, carbon dioxide is going to enter the leaf through the openings called stomata, all right? So if we look at the image here, the opening itself is the stomata, right? The actual opening. The cells surrounding that opening are not part of the stomata. They actually have a name that we call guard cells that open and close the stomata. But the stomata is the actual pore, it's the actual hole itself. All right, so carbon dioxide is going to enter. Through photosynthesis, we're going to produce oxygen. We have to have a way to release that oxygen. So oxygen will be released through those stomatas, through those pores. All right, we're going to go a little bit deeper into the anatomy here. All right. So if we're looking at our cross-section of our leaf here, and we zoom in on just a single plant cell, right? So one of those mesophyll cells. If we zoom in even further, we have that specific organelle, the chloroplast, right? The chloroplast is the organelle that carries out photosynthesis. We have talked about this in the first unit. But within these chloroplasts, there are pigments that are embedded in the membranes of our chloroplasts that are called chlorophyll. Right? So don't get those two names mixed up. Chloroplast is the organelle. Chlorophyll is a molecule embedded in the membranes of our thylakoids here, which we'll talk about in the next slide. Right, so if we look at the membranes, all of these little green dots that are clustered together, these are chlorophyll molecules. The chlorophyll molecule itself consists of this head and a tail, right? Has a very similar structure to maybe like a phospholipid, right? That also has a head but two tails, right? So they are embedded within this membrane. All right, it's chlorophyll that we will see play a really important role in collecting the sunlight. All right, I mentioned it was embedded in a membrane. So if we think about chloroplasts, we kind of talked about the anatomy in Unit 1, right, where it has this double membrane system because of the endosymbiotic theory, right? So it has an outer membrane, it has a space, and then it has this inner membrane, right? But chloroplasts have a third membrane that we call the thylakoid membranes, right? So these thylakoids, they look like pancakes that are in stacks to me, right? They consist of membranes in which the chlorophyll pigments are embedded. If we stack them all together, right, so a stack of thylakoids, we would call that the granum. So just a ton of membranes. It's like this membrane system inside of our chloroplasts. If we look at beyond a membrane and talk about what's surrounding all of these membranes, or what's inside of the membranes, we would say the stroma is the region between the thylakoid membrane and the inner membrane, right? So it's the region surrounding all of our thylakoids. If we were to look inside of a thylakoid, that space we would call the lumen, right? And the reason I mentioned these specific names, because when we talk about the light reactions, you will often see diagrams where there's a membrane, and on one side is the stroma, on one side is the lumen, right? So it's important that you know what they are. Stroma is outside of the thylakoids, right? Lumen is inside of the thylakoids. I like this image at the bottom here because it shows a non-cartoon version of a chloroplast. So if we were to actually look at one under a high-power microscope, we would see something like this green bean-like image with these areas of darker stacks. Those darker stacks would be stacks of our thylakoids. They're really membrane dense. All right, so I threw out a lot of terms at you. Let's review these terms. So in what cell types does photosynthesis take place? Where do we find the chlorophyll molecules? And then I want you to label the anatomy of this image up here. Ignore number 6. We did not talk about that one there.
Here's number 1 pointing to.
Number 1 is pointing to a membrane on the inside.
The third one? Third membrane?
Not the third one.
Second one?
The second one. Yep. Circle 1 is for transportation. Yes. Yeah. Well, it's actually going from this one to CO2. Yep. But circle to the right is a which one?
The original one and then it changes, or the one that it changed to?
I see what you're saying. Well, I always think in terms of that's what's changing in the reaction. The products, and that's the reactants.
So like the first,
Yes,
the first one is getting on.
They're the ones that actually changing.
and then a new one. You guys will watch.
Is it because like this one is
With the hydrogen, will it be one?
and this one
Yeah.
But if you have these this made sense to me too, because if the reaction went the other way, then this one would be the oxidized. Right? Because it's going
I always have to say that in mind. Moving. Yes, it would be easier to do one.
like if it was going this way, and these were the first ones, then yes, it would be this one. Yes, it goes to this. But since we're going this way, it's yeah. And then what would you call it? That doesn't have H. Like if the
You could
first
say it has been oxidized or has been reduced, I guess. I mean, as long as I am seeing that you're understanding what you're going for. Yeah. All right, so in what cell type does photosynthesis occur? Starts with an M. There are two different types of it. We're very quiet for a Friday. I have to get up and do jumping jacks or something. It's the mesophyll cells. Yeah, mesophyll cells is where this is occurring. Thought I had the answer up there. Right? Where do we find the chlorophyll molecules then?
starts with T, right?
Starts with a T. Yeah, membrane of the thylakoids. They are embedded in those thylakoid membranes. All right, if we were to look at this diagram, let's start with number 3 because it's the outermost part of our organelle here. What would you label number 3 as? Outer membrane, which would mean number 2 is? It's the inner membrane space as we're working our way inwards, which means number 1 would be that inner membrane, right? Then hopefully you got the rest of them here, right? So number 4 is stroma. It's just the area between our thylakoid membranes and our inner membrane, right? Number 5 is a thylakoid. And then number 6 is something called the lamella. We did not talk about it, but it's basically a bridge between stacks of thylakoids. All right, any question about the anatomy? I love talking about plant anatomy, so I wish I could do a whole lecture dedicated to this. All right, so all of this anatomy leads to photosynthesis. This is where photosynthesis is taking place. Photosynthesis can be broken down into 2 stages. Right? Just like cellular respiration was broken down into 4 stages, photosynthesis is broken down into 2. We have the light reactions, named so because we need the sunlight for these reactions. These reactions produce energy, produce ATP. They produce oxygen. They also produce this molecule NADPH, which is very similar to NADH. Right? It acts in the exact same way. It is an electron carrier. So we have our light reactions, but then we have what's called the Kelvin cycle. Often it's called the dark reactions because you don't need sunlight for it to occur. The Kelvin cycle uses what's produced in the light reactions to make glucose. Right? So it uses the ATP and NADH from the light reactions as well as some CO2, right, to produce glucose molecules. Glucose molecules that can then be used for cellular respiration in other parts of the plant. Today we're going to focus on the light reactions, right? We will save the Kelvin reaction or the Kelvin cycle for Monday. I think the Kelvin cycle is a little bit easier to learn, so let's start with the hard part of it first. we're going to start with the light reactions. Now, in order to understand the light reactions, we have to understand a few fundamentals about light itself. Right? So light is a type of radiation, electromagnetic radiation. It's going to travel as waves, and those waves can vary from short to long wavelengths. So if we look at what we call the visible spectrum, what we as humans can see with our eyes, right, we have a whole variety of colors. And within this visible spectrum, we get wavelengths that are closer together and then wavelengths that are really long and spread apart. All right, so they're traveling as waves, and those wavelengths can vary. But light also acts as a particle, right? And we call those particles photons. So we could say the photons of light are being absorbed, or the photons of light are hitting this surface. Right? So they move as waves, but they act as particles called photons, very discrete packages of light. Now, depending on the type of light we're looking at, right, so blue light tends to have a closer together wavelength, shorter wavelengths than red light. Shorter wavelengths carry more energy. Right, and we will see the significance of that very shortly here. But the wavelengths that are shorter, more compact, closer together, have higher amounts of energy. All right, so we have our particles, we have our photons. They are traveling in our waves. What happens to those photons when they interact with a surface? All right. Well, photons can do one of three things. They can be absorbed, they can be transmitted, or they can be reflected. All right, so I like this image up here because we see all of that happening here. We see light coming in. We can think of this light symbol here as a photon. The photon is striking the chloroplast. It can either be reflected off, right? So it just kind of bounces off the surface, goes in a different direction. It can be transmitted, so it might hit the thylakoids and just go right through them, right? They're just traveling through. They're being transmitted through. Or they can be absorbed, right? Where the light will hit those thylakoids and something happens to those photons, right? They're being absorbed and being captured, used as a source of energy. And what's actually capturing and absorbing these photons are the pigments, right? We talked about chlorophyll. Chlorophyll being embedded within our thylakoid membranes here, right? Chlorophyll is a pigment, and a pigment is a molecule that will absorb only certain wavelengths of light. Remember, we have all of this visible light, right? Ranging from blue light to red light, right? All the different wavelengths in between. Pigments are designed to pick up on just a certain segment of that visible light, to capture only just a sliver of it. Right? So for example, when we think about plants, right? We know they're green. Most plants are green. There are always exceptions, right? Leaves are green because what they are doing is absorbing the red light. They're absorbing the blue light because these are the light wavelengths that the pigments themselves will actually absorb. But these pigments do not absorb green light. That light is being reflected back at us, which is why we see the leaves as green. Because the leaves aren't using green, it's being reflected out, being reflected back to our eyes. We see leaves as being green.
Why do they change colors?
Ooh, great question. We'll get to that. Yes, so right now you're thinking about all those fall trees out there and why some of them aren't green. We will talk about that. We're going to circle back to that question. All right, so we have photons, right? They can be transmitted, they can be reflected, or they can be absorbed in pigments. What happens when they are absorbed by a pigment? Well, they can do, again, a variety of a few different things. They can, well, let me back up. when it's absorbed by a pigment, right, we actually excite the electrons, right? An electron is being excited in the chlorophyll pigment themselves. So when I say an electron is being excited, right, remember our atom, we have these different electron shells. If we have an electron in an electron shell close to the nucleus, right, it's in a ground state, very stable. But if we excite this electron, right, so a photon of light hits this electron can become excited and jump to the next electron shell out, right? We would call this an excited state, right? This is not a stable state for the atom, right? It's very unstable. Unstable to the point where this electron really doesn't want to be there. It wants to go back down to its ground state. It wants to become unexcited, right? So this electron has all of this energy that it has to lose. Doesn't like being excited, right? It's because it's not stable when it's excited. So it has to lose this energy, and this energy can be lost in a few different ways, right? It can be lost as heat, right? So if you think about if you're driving or walking in a parking lot and you see like the heat radiating off the blacktop, right? It can be lost in that type of radiation. It can be lost as light, right? So all plants actually do this. They fluoresce, right? Or they emit light, just like these jellyfish here. But plants do it so minimally that we can't see with our naked eye, right? We have to have special instrumentation to see this. Unlike our jellyfish, which, I don't know, has anybody seen biofluorescent jellyfish before? I think that'd be really cool to see. Yeah. Yeah, like a zoo or something, yeah. So they emit all that excited energy as a light, right? But what plants do in photosynthesis is that excited energy is being transferred to another molecule, right? So it's being captured by something else. So once our electron loses that energy, right, that energy is being captured by another molecule. All right, so we have our photons of light. They can be absorbed, transmitted, or reflected. In photosynthesis, they are absorbed, right? When the pigment absorbs that light, right, we're exciting an electron, and we have to get rid of that excited energy. We can do that via heat, light, or transferring that energy to another molecule. So in photosynthesis, we're transferring that energy to another molecule, all right? So here we have our chlorophyll pigment, right? The head of the chlorophyll pigment is where that light is going to be absorbed and where that electron is going to be excited, right? It's going to raise that electron to a higher energy state. But if remember, light is not all the same. We have this spectrum of visible light, right? Blue light has really short wavelengths. Red light has really long wavelengths. They carry different amounts of energy, right? So if we see these photons hitting an electron, right? I like this graph. It gives a really nice visual representation, right? The blue light has shorter wavelengths. See how it's more squiggly in this line here. So the wavelengths are closer together. It's going to excite the electron to a higher energy state simply because blue light carries more energy. Whereas red light is a shorter wavelength, still going to excite the electron, but not as much, right? Because it carries less energy with it. So if you've ever driven past a greenhouse at night and maybe you see like the greenhouse looks purple, right? The lighting's all purple. That's because the greenhouse is giving the plants blue and red light, which look purple when combined. Plants actually prefer blue and red light to all of our spectrums because they can't utilize green. So to be as efficient as possible, greenhouses will only provide green and red light. Blue and red light. All right, that electron is in that excited state, and we're going to lose that energy, that excited energy, by transferring that energy to another molecule. So, chlorophyll pigments, they have their heads, they have their tails. They often are in groups that we would call an antenna complex. We don't see an isolated pigment here or there. Rather, we see clusters of 200 to 300 pigments all working together, right? So we call this the antenna complex. We'll find them embedded within our membranes here. Now we know that this electron can get rid of its energy by fluorescing or losing heat. And in plants, they do this to a small degree. But most of that energy is going to be transferred to another molecule. So we would call this resonance energy transfer, right? So if we look at our image here, we have a photon hitting a chlorophyll pigment. So one of these jagged green slices here are our chlorophyll pigments. The energy is hitting that electron. That electron is being excited. What's going to happen is this electron itself is going to bounce between different pigments, right? So it's going to go from pigment to pigment to pigment to pigment until it reaches the center of our antenna complex, until it reaches the reaction center. The reaction center is the last stop for this electron, right? So it reaches that reaction center. That electron is going to be transferred to an electron acceptor, right? So we have to capture that electron. It's going to go to an electron acceptor. All right, so we have all these pigments working together, working as one in these giant groups of 200 to 300 molecules. Our energy in our electron is bouncing from pigment to pigment to pigment until it reaches the reaction center, in which point it's going to be given to an electron receptor. There are other pigments besides chlorophyll, all right? So especially now, we're looking outside at the trees. The trees are not all green anymore, right? So we often talk about chlorophyll because they're the main pigment that will accept the photons. But there are other pigments that we would call accessory pigments. And these are things like carotenoids and xanthophylls. They tend to show up as these reds, these yellows, these greens, right? What they do is they just absorb light in a slightly different wavelength. Right? So chlorophyll, they cannot absorb any green light. These carotenoids and these xanthophylls, these accessory pigments, they can. They can extend the amount of light that can be absorbed. So we see these carotenoids here have high light absorption even when that light is green. There are other pigments that basically kind of act like a sunscreen for the plant, right? So the plant's not going to get too much energy. So in the fall time, what happens is that these plants are getting ready for winter, right? They're going to become dormant, essentially. All of the chlorophyll pigments are being broken down and brought back into the tree itself so that those pigments can be recycled into other molecules. What remains in the leaves are these accessory pigments. So most of the time, they're overshadowed by the abundance of chlorophyll, but in the fall, right, when it's all going back into the tree itself, what's left is these accessory pigments. And that's why we see these bright, colorful trees in the fall. All right, I realize a lot of information, all right? So let's do a review, all right? What are 3 ways that an electron can release its captured energy? Does red or blue light excite an electron more? And then there's this plant that we call red algae, right? This plant appears red to the naked eye. Why is that so? And what wavelengths of colors does this light use to photosynthesize? So I want you to give these some thought. And talk with one another.
Fridays I have like very energetic classes. I
I would assume it has something to do with the year that they've had. Like maybe it was like especially hot and sunny, they might produce more accessory pigments that kind of protect against too much damage from the sunlight.
So that the chlorophyll
I did you? Oh, they were pretty incredible. Yeah. It's only the second time I've seen them. I know. Right. Something about the sun or something, or we're getting bored. Yeah.
I have a question.
Yeah.
So red algae, would the red algae be with the
Yes, so red does have a long wavelength. Why are we seeing it? Is it being used by the people? But why was it being used? Basically, and they're emitting and reflecting green back at us. All my faces now are back the same value, red wavelengths, and emits it back to us. What type of wavelengths does it want?
That's what I thought. I just haven't started about it.
All right. What are 3 ways that an electron can lose its captured excited energy? Heat, light,
The
it can
jellyfish.
transfer it.
Oh.
Yes. So heat, light, or transfer it. Does red or blue light excite an electron more?
Blue.
Blue. Why is that?
Because the waves are shorter and it's brighter.
Yeah, shorter wavelengths. Shorter wavelengths have more energy. And then finally, we have this red algae plant that we find in like marine waters. This plant appears red. Why do we see it as red? Yeah, because it's the red being reflected out. We see trees as green because it's reflecting the green back at us because it's not using the green. We see this algae as red because it's reflecting the red back at us. It's not using the red. So what colors of light does this plant use to photosynthesize? Yeah, blue and greens, right? So it utilizes slightly different pigments that can maximize on the blues and the green light that it's receiving instead. Any questions so far? All right. So now we have a little bit of a background as to why light has energy. Right? Let's look at specifically how this energy, once it excites an electron, once that energy has been captured, powers the light reactions. Right? So again, we're looking at the thylakoid membranes inside of our chloroplast. This is where our light reactions are occurring. We're using that light energy. We're also using water, right? And we're going to create oxygen. We're going to create ATP, and we're going to create NADPH, right? NADPH is just like NADH, right? Just a slightly different variation on that molecule, right? It is an electron carrier. It carries that energy. Often when I teach this unit, I tend to get NADH and NADPH mixed up so much because we talk about both in different ways. right? But NADPH, think P, photosynthesis, right? So NADPH is associated with plants. All right, so we have that electron. It's captured this energy, right? And it's at an electron-carrying molecule, right? This electron is going to go through a series of reactions, right? So when we talk about the light reactions, we are really talking about the movement of our electron that has been excited, right? We're going to break down this photo into step-by-step processes here, but essentially, this electron is going to move between different proteins in our thylakoid membrane, right? As it moves, similar to the electron transport chain, we're going to build up a gradient of hydrogens that will power ATP synthase. All right, so it should be slightly familiar in the big picture of what it does. All right, but we're going to break this down. So our thylakoid membrane, right? They have these chloroplast complexes, and they have two distinct complexes of them, right? We would call them photosystem 1 and photosystem 2, right? Now, the confusing bit is that they were named in the order in which they were discovered, right? So photosystem 1 was discovered first, photosystem 2 was discovered second, but it's actually photosystem 2 that's the first step in photosynthesis. So they were discovered backwards from how they're actually working in photosynthesis, right? So photosystem 2 happens first, then it moves on to photosystem 1. In these photosystems, this is where we have light exciting those pigments. This is where those photons are striking the pigments, exciting the electrons. The electrons are bouncing between pigment and pigment and pigment until it's finally captured. So we have these two different photosystems. If we look at a photosystem, right, it's going to contain two different main components. The antenna complex, which is all of those pigments working together, those clumps of like 200 to 300 pigments. They're directly absorbing the photons, right? So the photon comes in, hits that electron becomes excited, bounces between pigment to pigment to pigment, until it reaches that other bit of our antenna complex that we call the reaction center. It's the reaction center that differs between photosystem 1 and photosystem 2. Right? So the reaction center, right, kind of you can think of it as like the middle of this complex. We would call it P680 in photosystem 2 and P700 in photosystem 1. That refers to the specific wavelength that it captures the best. Right? It doesn't only capture that wavelength, right? But it captures that wavelength the best. That is the only difference between these two photosystems, right? When I put an asterisk behind the reaction center, right, so you can see P680 asterisk, that is indicating that it's been excited, right? It has captured an excited electron. And we know that excited electron carries a lot of like chaotic energy that it needs to get rid of so it can become stable again. So that energy is going to be transferred pretty quickly. And it's this reaction center that will take that chaotic electron and give it away to an electron receptor. I'm going to be repeating the movement of this electron quite a few times, right, to really reinforce this idea. So we have our antenna complex, lots of pigments, the reaction center's in the middle. It gives away that excited electron to a different molecule. All right, so we have pigments in the middle here, embedded in a membrane. I want you to answer these three questions.
Like, can you put it outside or like the top floor?
But then it leads to outside.
Happening in the sky.
All right, what is the name of this cluster of pigment molecules?
Something complex.
Something complex.
Just an A.
Antenna complex. Yes, it's the antenna complex. So if a photon were to strike this yellow pigment molecule, in what manner is it going to travel to the reaction center, or this red dot?
Hop.
Yeah, it's going to hop from pigment to pigment. So if you were actually drawing this path on here, there's many different ways you could draw it, right? It's going to hop from pigment to pigment to pigment until it reaches the reaction center. It doesn't have to take this path. It might take a different path. Yeah.
Would that like specifically be called like resonance or something like that? Or?
Yes, my last question, the name of this type of travel. Resonance energy transfer. Yes. So this is what we call a resonance energy transfer when it bounces from pigment to pigment to pigment. All right. Any questions so far? Like I said, the light reactions are the harder of the two stages of photosynthesis. Just because there's a lot going on. All right, so like I mentioned, photosystem 2, even though it's number 2, it happens first in our light reactions, right? This is technically the first step in photosynthesis, right? Light is going to excite an electron in this photosystem, in this antenna complex. And it's going to release that energy, right? So that electron that's excited is bouncing from pigment to pigment to pigment to reaction center, being accepted by a molecule. Right? Once it's accepted by that molecule, at the end of photosystem 2, it's going to move on to an electron transport chain. Right? So photosystem 2 is where we're actually capturing the photon of light and exciting this electron. Right? I like this image because it gives a nice clear picture of what's going on. We have our light. It's being absorbed by one of our pigments. Right? It's bouncing back and forth until it reaches the reaction center. Now the reaction center in photosystem 2 is called P680. Right? It's going to be really unstable when it gets this energy. So it's going to get rid of this energy really quickly. And it's going to give that electron away with its energy to a primary electron acceptor. Right? Now, the reaction center is stable. Right? It's been accepted by an electron acceptor, which makes it relatively stable. Right? The problem is, we've given away an electron. Right? If we keep giving away electrons, there's going to be no more electrons to excite. So we have to gain an electron back into the system. This is where water comes into play. All right? We are going to split water, and by the splitting of water, we're giving away an electron from our water molecule back into our photosystem. And in the process, we're creating oxygen. Right? Some hydrogen ions and oxygen. So light's coming in, it's bouncing that electron from pigment to pigment. It reaches the reaction center. That's very unstable, so it's going to give that excited electron away to a primary electron acceptor. But now we need to have another electron come in to replace the one we lost, and we get that when water is split apart. Right? This is very unique in plants. All right? This is called oxygenic photosynthesis, and photosystem 2 is the only protein complex that's able to split apart water in this manner. Right? So we generate oxygen. We also generate some ions. We give away that electron from water, right, to replace the one that we lost in our photosystem. We're also generating some hydrogen ions from splitting of water, which will eventually go on to make ATP, right? Like I said, this is very unique in plants. I've designed this lecture so I repeat myself many times because it's a hard concept to grasp for many. So again, this slide here is just repeating that splitting of water. All right. As we go through the light reactions, we're going to fill out this graph. So here we have a graph, right? We have the movement of electrons through the light reactions on the x-axis and the energy level of the electrons on the y-axis. Okay? I want you to draw an arrow up or down for photosystem 2 as to how the energy level of this electron is going to change. And then I want you to tell me, what are the products of photosystem 2? What does it produce? I'll give you a couple minutes. You all are making me feel really tired, just the low amount of energy in this room. If we were to look at this graph, right, we have light hitting this electron. Is the energy level going to increase or decrease? I think I heard the whispers. It's going to increase, right? So the energy level is going to increase. And we think about photosystem 2, what is being produced here? Oxygen. Yeah, we're producing oxygen. This is the step that allowed for oxygen-breathing organisms to evolve, right? What else is being produced?
H plus.
Yeah, some protons, some hydrogen ions, right? So we're producing oxygen and hydrogen ions. Where did these come from? What molecule? Water, yeah, H2O. All right. That electron, it's been excited. It's been given to a primary electron acceptor. It's now going to go through an electron transport chain. All right, so electrons are going to leave photosystem 2. It's going to travel along the electron transport chain until it reaches photosystem 1, right? This is a series of molecules embedded in the membrane, right? Series of reactions. As this electron moves, very similar to the electron transport chain in cellular respiration, it provides energy by which hydrogen ions are transported across the membrane, building up a gradient, right? And this gradient will ultimately be used to make ATP. Right, so we've gone from photosystem 1 to an electron transport chain. As this electron moves, right, it's providing energy for the movement of these hydrogen ions, which means the electron itself is losing amounts of energy. Right, it's giving some of this energy away to power this movement. So if we were to come back to this graph, what is it going to look like when we add an arrow? And we're going to do this diagonally, right? Is this electron going to gain or lose energy, right? And then what is happening during the electron transport chain? We didn't talk about too much about it because it acts very similar to how in cellular respiration. All right, here, what do we have planned for the weekend? Oh, sure. Yeah, that's fun. Very close to where I live. I should maybe try to go this year. I saw a hand up. Was your question?
Oh, no, I was just drawing.
Oh, which one? Arrow, perfect. All right, I think I'm going to cut this question short so we can keep moving here. If we were drawing an arrow, right, is this electron going to gain or lose energy? It's going to lose energy, yeah. So during the electron transport chain, it loses energy, right? And what's happening during the electron transport chain? What is this electron providing energy for? Very similar to what it provides energy for in cellular respiration. Yeah, it's not just the electron moving, right? It's also building up that gradient of hydrogen ions in order to make ATP eventually, right? So hydrogen ions are being pumped across the membrane, being used to make ATP. All right. This means our electron has now reached photosystem 1, right? Photosystem 1, again, is a complex of pigments with a reaction center. It's not gaining an electron from water. Rather, it's just receiving the electron that has just gone through the electron transport chain. We know this electron has lost a lot of energy. So what photosystem 1 does is it re-excites the electron, right? So another photon of light comes in, hits the electron, and re-excites that electron, gives it even more energy, right? It's the same process as before, it just uses a slightly different reaction center. So again, that P700 versus P680. Right, so we are re-exciting that very same electron that we have been talking about in the last few systems here. Now, once this electron is excited, its primary role is to make NADPH. Right, so this electron, once it's given away to its electron scepter from the reaction center of photosystem 1, it wants to make NADPH. So it's been re-excited in photosystem 1. It leaves photosystem 1. It again goes through a series of reactions. It goes through basically another electron transfer chain, right? Series of reactions until it reaches NADP+. NADP+ will finally accept that electron. It is the last electron acceptor to make NADPH, right? An energy-containing molecule. Right? And that's what this whole process is about. Creating NADPH and setting up a gradient of hydrogen ions. That's the whole reason for these light reactions. All right, let's talk through this last one together, and then we will finish up on the light reactions on Monday. Right? So we have our electron, photosystem 2, it excited it, right? It lost some energy through the first electron transport chain, right? We now have it hitting another photon of light. What is happening to this electron? Yeah, it's being re-excited, right? Re-excited, so it's going to a higher energy state, okay? And then once it's re-excited, it's going through another electron transport chain. Is it going to lose or gain more energy? Just like before, it's going to lose energy until it finally gets accepted by NADP+ to make NADPH. Right? That is the last step. All right. I have a content check due for Monday. I'm going to reevaluate that. I think you should still be able to get it done for Monday. But if we run into problems, I might extend that by another day. Right? Otherwise, Monday, think about the next content check. Scientific literacy assignment on Wednesday, exam on Friday.